Hi everyone, welcome back to the Science Box. So today I'm going to be telling you everything you need to know for the exchange chapter of AQA A Level Biology. The timeline's in the description, so if there's a particular part you're stuck on, feel free to just skip to there. And let's get started. So as we know, organisms exchange multiple things with their environment, including respiratory gases, so oxygen and carbon dioxide, nutrients, so glucose, fatty acids, amino acids, vitamins, minerals. They exchange excretory products, so urea and carbon dioxide, and then they also exchange heat. I've just put there, in an exam, don't use the word nutrient, specify the type of nutrient you're talking about because it could be a huge number of things. Except for heat, the exchange of these substances happens passively by diffusion or osmosis, or actively by active transport. The larger the organism, the smaller the surface area to volume ratio. Therefore, there's less area for a substance to diffuse compared to the number of cells inside that need that substance. You may be asked to calculate the surface area to volume ratio in your exam, so I've put 2 examples here for you to try. Feel free to skip them if you're confident, but definitely pause and try them if not. Okay, so for this first question, we first need to find the surface area of the cylinder using the formula, and then we're going to find the volume. And if we make those a ratio, so it would be 113 to 87, we can then divide both sides by 113 to get a ratio which is 1 to whatever. So in this case, 1 to 0.778 to 3 significant figures, which would be our final answer. On the right, things get a bit more complicated. I'd say the main thing is to remember that the bottom disc of that cylinder isn't included in the surface area. So you're only going to count the things that are kind of exposed to the air, if you like, exposed to the outside. So you can see for the surface area of the cuboid, I've minused that little disc, the pi r squared. And then surface area of the cylinder, I've done the same. I've only done one disc instead of the 2. We're going to add both the surface areas together, both the volumes together. The answer becomes 424 to 460, and if we divide those both by 424.53, we get 1 to 1.08 to 3 significant figures. Okay, so even if the outer surface could supply enough of a substance, it would still take too long for it to reach the middle of the organism if diffusion alone was the method of transport. This is why larger organisms have specialised gas exchange surfaces, like gills or lungs, or sometimes have a flattened shape, so no cell is ever far from the surface, like flatworms do. Specialised exchange surfaces show the following characteristics. A large surface area relative to the volume of the organism, which increases the rate of exchange. Very thin, so that the diffusion distance is short and therefore materials cross the exchange surface rapidly. Selectively permeable to allow selected materials to cross. Movement of the environmental medium, for example air, to maintain a diffusion gradient. And a transport system to ensure the movement of the internal medium, for example blood, in order to maintain a diffusion gradient. The rate of diffusion is proportional to the surface area times the difference in concentration all over the length of the diffusion pathway. This is Fick's law. Single-celled organisms exchange gases by passive diffusion since they have a large surface area to volume ratio, but insects have a more complex gas exchange system. Insects have an internal network of tubes called tracheae. You may have heard that word from studying the lungs because the main pipe leading to the lungs is the trachea. Anyway, the tracheae are supported by strengthened rings to stop them collapsing. The tracheae also divide into smaller dead-end tubes called tracheoles. In this way, atmospheric air is brought directly to respiring tissues as there's a short diffusion pathway. Respiratory gases move in and out of the tracheal system in 3 ways. Firstly, along the diffusion gradient. Oxygen is used up by respiring cells, creating the diffusion gradient, and carbon dioxide produced by respiring cells diffuses in the opposite direction. With mass transport, muscles in the insect contract, squeezing the tracheae and enabling mass movement of air in and out, speeding up the exchange of respiratory gases. The ends of the tracheoles are also filled with water. During periods of major activity, muscle cells around the tracheoles respire anaerobically, producing lactate. Lactate lowers the water potential of the muscle cells, so water moves into the cells from the tracheoles via osmosis. The water in the ends of the tracheoles decreases in volume, drawing air further into the tracheoles. This means the final diffusion pathway is in gas phase rather than liquid phase, so diffusion happens more quickly. Gases enter and leave the tracheae through tiny pores called spiracles on the body's surface. The spiracles open and close by valves, and when open, the water vapour can evaporate from the insect, so they're usually kept closed. Now, fish have a very efficient gas exchange system to obtain the maximum amount of oxygen from the water into the blood. Fish have gills, and you can see a diagram of a gill on the screen there. Each gill is made of gill filaments, so each one of those is a gill filament, and on these filaments are lamellae. The lamellae are at right angles to the filaments, as you can see here. Water is taken in through the mouth and pushed over the gills in the direction you can see on the diagram, so at a right angle to the filaments. Both the filaments and the lamellae provide a large surface area for diffusion, and the lamellae are thin, providing a short diffusion pathway. The lamellae have a really good blood supply as well, which maintains the concentration gradient. To fully maximise the amount of oxygen that diffuses from the water into the blood, fish use something called the countercurrent exchange principle. This is all based off of the blood and water flowing in opposite directions. This means the blood, already well loaded with oxygen, meets the water which has its maximum concentration of oxygen, so diffusion of oxygen from water to blood still takes place. That's shown in this diagram here, where the percentage of oxygen is always higher in the water than in the blood, so oxygen always diffuses into the blood. The blood with little oxygen meets water which has had most of its oxygen removed, so diffusion of oxygen from water to blood still takes place. In other words, the diffusion gradient for oxygen uptake is maintained across the entire width of the gill lamellae. Now moving on to gas exchange in plants. Plants both photosynthesize and respire, meaning the balance of gases diffusing in and out of the leaves varies. Plants in general take in carbon dioxide through diffusion and diffuse out oxygen. But during the night, for example, when photosynthesis isn't taking place, plants will take in oxygen and diffuse out carbon dioxide. The plant is adapted for gas exchange in the following ways. The leaves have many small pores, called stomata. No cell in the leaf is far from a stoma, and therefore the diffusion pathway is short. The leaves have numerous interconnecting air spaces that occur throughout the mesophyll, so that gases can readily come in contact with the mesophyll cells. And the leaves have a large surface area of mesophyll cells for rapid diffusion. Additionally, diffusion takes place in the gas phase, which means it's more rapid than if it were in water. Each stoma is surrounded by a pair of guard cells that open and close it. This is useful for gas exchange, but also limiting water loss. In exchanging gases with the environment, a lot of organisms suffer with water loss, so they have adaptations to deal with this. Insects have a small surface area to volume ratio. They have waterproof coverings over their body's surface, and spiracles that can close. Certain plants are called xerophytes, which means they're adapted to living in areas where there's little water by limiting water loss. They may have a thick waxy cuticle that forms a waterproof barrier, or they may roll up their leaves, which traps a region of still air. This region becomes saturated with water vapour, so has a very high water potential, so there's no water potential gradient, and so no water loss. They may have hairy leaves which trap moist air next to the leaf's surface. The water potential gradient between the inside and the outside of the leaves is reduced, therefore less water is lost by evaporation. The stomata can be in pits or grooves to trap moist air next to the leaf and reduce the water potential gradient. And finally, they have a small surface area to volume ratio, decreasing the area over which water can be lost. Now let's move on to the human gas exchange system. Here are the main parts of it. The trachea is a flexible airway that is supported by rings of cartilage. The cartilage prevents the trachea collapsing as the air pressure inside falls when breathing in. The tracheal walls are made up of muscle lined with ciliated epithelium and goblet cells. The bronchi are two divisions of the trachea, each leading to one lung. They are similar in structure to the trachea and, like the trachea, they also produce mucus to trap dirt particles and have cilia that move the dirt-laden mucus towards the throat. The bronchioles are a series of branching subdivisions of the bronchi. Their walls are made of muscle lined with epithelial cells. This muscle allows them to constrict so that they can control the flow of air in and out of the alveoli. The alveoli are minute air sacs at the end of the bronchioles. They're lined with epithelial cells. The diffusion of gases between the alveoli and the blood will be very rapid because red blood cells are slowed as they pass through the pulmonary capillaries, allowing more time for diffusion. The distance between the alveolar air and the red blood cells is reduced as the red blood cells are flattened against the capillary walls. The walls of both alveoli and capillaries are very thin, and therefore the distance over which diffusion takes place is very short. Alveoli and pulmonary capillaries have a very large total surface area. Breathing movements constantly ventilate the lungs, and the action of the heart constantly circulates the blood around the alveoli. Together, these ensure that a steep concentration gradient of the gases to be exchanged is maintained. And finally, blood flow through the pulmonary capillaries maintains a concentration gradient. The breathing movement is split up into inspiration and expiration. In inspiration, the external intercostal muscles contract and the internal intercostal muscles relax. The ribs are pulled upwards and outwards, increasing the volume of the thorax. The diaphragm muscles contract, causing it to flatten, increasing the volume of the thorax. The increased volume of the thorax results in reduction of pressure in the lungs, and the atmospheric pressure is now greater than the pulmonary pressure, so air is forced into the lungs. Expiration is going to be the exact opposite, so the internal intercostal muscles contract and the external intercostal muscles relax. The ribs move downwards and inwards, decreasing the volume of the thorax, and the diaphragm muscles relax so it resumes its dome-like shape, decreasing the volume of the thorax. The decreased volume of the thorax results in an increase of pressure in the lungs. The pulmonary pressure is now greater than the atmospheric pressure, so air is forced out of the lungs. Like I said, those two are just opposites, so when you've memorised one, you've memorised the other. If you're struggling to remember which one is which, the way I do it is by working backwards. So with inspiration, for example, I know that the end effect is that air goes into the lungs. So for that to happen, the atmospheric pressure has to be greater than the pulmonary pressure, so air is forced into the lungs. If we want the pressure inside the lungs to be low, then we want the volume to be high. So we're going to think, what's going to make the volume higher within the lungs? We're going to pull the ribs upwards and outwards, and the diaphragm we want to flatten, so there's a larger volume within the lungs there. So yeah, that would be my tip, just work backwards. Now moving on to enzymes and digestion. Here are the main parts of the digestive system. The salivary glands pass secretions containing the enzyme amylase, which hydrolyzes starch into maltose. The esophagus carries food from the mouth to the stomach. The stomach physically digests the food and has peptidase enzymes which digest proteins. The pancreas is a large gland situated below the stomach. It produces a secretion containing proteases to hydrolyze proteins. lipases to hydrolyze lipids, and amylase to hydrolyze starch. Food is further digested in the ileum by enzymes that are produced by its walls and by glands that pour their secretions into it. The inner walls of the ileum are folded into villi, which give them a large surface area. The surface area of these villi is further increased by millions of tiny projections called microvilli on the epithelial cells of each villus. This adapts the ileum for its purpose of absorbing the products of digestion into the bloodstream. The large intestine absorbs water, and the rectum is the final section of the intestines. The feces are stored here before periodically being removed via the anus in a process called digestion. In humans and many other organisms, digestion takes place in two stages. First, it's physically broken down, and then it's chemically digested. The physical breakdown of the food provides a larger surface area for enzyme action during chemical digestion. The chemical digestion of carbohydrates, proteins, and lipids each happen slightly differently. With carbohydrates, polysaccharides like starch are first broken down into disaccharides by one enzyme, and from disaccharides to monosaccharides by another enzyme. First, the food is chewed and saliva is produced from the salivary glands. Saliva contains salivary amylase. Amylase hydrolyzes alternate glycosidic bonds of the starch molecules to produce the disaccharide maltose. It also contains mineral salts that help to maintain the pH at around neutral. This is the optimum pH for salivary amylase to work. The food is swallowed and enters the stomach, where the conditions are acidic. This acid denatures the amylase and prevents further hydrolysis of the starch. After a time, the food is passed into the small intestine, where it mixes with the secretion from the pancreas that contains pancreatic amylase. This continues the hydrolysis of any remaining starch to maltose. In the ileum, the epithelial lining produces the disaccharide maltase. Maltase is not released into the lumen of the ileum but is part of the cell surface membranes of the epithelial cells that line the ileum. It is therefore referred to as a membrane-bound disaccharidase. The maltase hydrolyzes the maltose from the starch breakdown into alpha glucose. In addition to the maltose, there are two other common disaccharides in the diet that are hydrolyzed: sucrose and lactose. Sucrose is found in many natural foods, especially fruits. Sucrase hydrolyzes the single glycosidic bonds in the sucrose molecule. This hydrolysis produces the two monosaccharides, glucose and fructose. Lactase hydrolyzes the single glycosidic bond in the lactose molecule. This hydrolysis produces the two monosaccharides, glucose and galactose. Proteins are digested by peptidases. We start off with a long peptide chain like this. Endopeptidases hydrolyze the peptide bonds between the amino acids in the central region of a protein molecule, forming a series of peptide molecules. Exopeptidases hydrolyze the peptide bonds on the terminal amino acids of the peptide molecules formed by endopeptidases. In this way, they progressively release dipeptides and single amino acids. Dipeptidases hydrolyze the bond between the two amino acids of a dipeptide. Dipeptidases are membrane-bound, being part of the cell surface membrane of the epithelial cells lining the ileum. Lipids are first emulsified by bile before being digested by lipase enzymes. This process happens as follows. First, the lipid droplets are emulsified by bile salts. This breaks down the lipids into smaller droplets, so there's a larger surface area for faster hydrolysis action by lipase enzymes. The lipase enzyme breaks down the lipids into monoglycerides and fatty acids. The monoglycerides and fatty acids remain in association with the bile salts to form structures called micelles. When micelles come into contact with the epithelial cells lining the villi of the ileum, they break down and release the fatty acids and monoglycerides. The fatty acids and monoglycerides diffuse across the cell surface membrane and are transported to the endoplasmic reticulum, where they're combined to form triglycerides. The triglycerides combine with cholesterol and lipoproteins in the Golgi apparatus to form chylomicrons. Chylomicrons move out of the cells by exocytosis. That brings us to the end of digestion, and so the end of the exchange chapter. Thank you for watching. Please like and subscribe. If you like this style of teaching and would like to get tutored by me or a member of our team, you can go to our tutoring website at thesciencebox.net. I'd also really recommend our Instagram and TikTok for tiny doses of revision. It makes you feel productive even if you're just scrolling TikTok, so that's a bonus.